All right, so we're going to understand today the steps and molecules required for RNA synthesis in humans, and it's much more complex than in bacteria. So we're going to have to go a little faster today. Let's try not to get too bogged down in the details and stick to the bigger picture, which is that there are a couple of different kinds of RNAs in the cell that are made by different RNA polymerases. Pol 1 makes the ribosomal RNAs. Pol 2 makes the messenger RNAs, which code for protein. And Pol 3 makes the transfer RNAs. And there are some other RNAs listed here that we won't spend a lot of time on, but we will mention their roles, the nucleolar RNAs, the small nuclear RNAs, in the process of making some of these other RNAs and in making protein. Now, the RNA that's made by Pol 2, the messenger RNA, is far more complex in humans than it is in bacteria. There are a lot of post-synthesis modifications that we're going to go through again pretty quickly: capping, tailing, splicing, editing. Okay? And then we're going to talk about, just introduce you to the idea that in eukaryotic genes, the promoter region is far more complex, and there are lots of players involved in regulating that. Tomorrow morning, Friday, we're going to really focus on that idea, on the control of mammalian gene expression, by really zooming in on promoters and promoter binding proteins. But we'll introduce that idea today. Okay, so everybody remembers from basic biology the central dogma. Information stored in DNA is eventually made into RNA, and that's eventually turned into proteins, and proteins then provide the structure and the function of a cell, tissue, organism. And again, to reiterate, eukaryotes are a lot more complex than bacteria. One of the key points here is there's compartmentation of these processes in human cells that does not occur in bacterial cells. Another concept that's not really illustrated but it's really important to understand is the RNA sequences and the proteins that catalyze some of these reactions are, in some cases, different, and sometimes profoundly different, sometimes subtly different between eukaryotes, prokaryotes. But in many cases, that is the principle for selective toxicity of really important drugs. Drugs used to treat infection, drugs used to treat cancer. Okay? And you'll want to know what those drugs are and how they work, because a lot of my exam questions are really oriented from the point of view of the pharmacology and how does the molecular biology explain it. Okay, so as I've already pointed out, the features of transcription are more complicated, lots more players in the eukaryote. So let's get down to it and talk about who does what, where, and how. So this is a summary table I think that'll help you keep a lot of this stuff organized. You'll want to study this table and know this. Okay? So Pol 1, again, ribosomal RNAs, and it's insensitive to this inhibitor alpha-amanitin. You go, well, what's that? Well, it's clinically not a terribly important compound. It is actually a toxin from poisonous mushrooms, and it's one of the classes of chemicals that kills people if they eat the wrong type of mushroom. Scientifically, it's important because it helps us discriminate between these different polymerases. And Pol 2, which makes messenger RNA leading to proteins, is strongly inhibited. So it's a transcriptional inhibitor that has been used experimentally to define what these polymerases do. And then Pol 3 is only weakly inhibited by this particular inhibitor. And as I said, it makes a different set of RNAs. Now, a minor point, but interesting one, is that there's also polymerases in mitochondria, okay? And they make messenger RNAs, ribosomal RNAs, and transfer RNAs, and they're not sensitive to the alpha-amanitin because they are actually quite different from these nuclear enzymes. And that is probably because these enzymes evolved from endosymbionts related to the Rickettsia bacteria that sometime in distant past became mitochondria. Okay. And because of that similarity or that evolution from bacterial proteins, the mitochondrial polymerases are, in fact, sensitive to the drug rifampin. We met rifampin yesterday, right? What's it used for? It's used for tuberculosis because it inhibits the RNA polymerase in microbacteria. And so some of the side effects from rifampin are probably due to inhibition of mitochondrial enzyme polymerases. Make sense? Okay. So that's an example of the kind of connections that I want you to be able to make as we go through these lectures. All right, so we'll just go in order, pole 1, 2, 3. I'll spend most of the time on pole 2. But pole 1 makes the ribosomal RNAs, except for one of the subunits called 5S. And remember that S means Svedberg units. It's kind of size, bigger number, bigger particle. So 5S is a little tiny piece of the ribosome. So where does all this happen? Well, in a cell, it happens in the nucleus, and particularly in the nucleolus. And there are a lot of ribonucleoproteins that are made in the nucleolus. So here's a EM of a cell showing the nucleus bounded by the nuclear membrane. And you see this density here. This is the nucleolus. Now what this is a collection of lots of pieces of about 10 different chromosomes in the human that all come together, and they coalesce along with a bunch of binding proteins and metabolic enzymes in this region. And the purpose is to make RNA and to assemble proteins that are important in the synthesis and the processing of RNA. And so you see these little green blobs here. This is meant to tell you that pieces of ribosomal RNA are synthesized in the nucleolus and then eventually complexed with proteins and then shipped out into the cell to form ribosomes on the endoplasmic reticulum. Okay, so they're originally made in the nucleolus, the various RNA that makes up these ribosomes. And in addition, there's a couple of other things made and assembled in the nucleolus. One we met the other day, right? The telomerase reverse transcriptase. And that's the enzyme that actually adds the telomeric repeats to the end of DNA. It actually gets its little piece of RNA in the nucleolus. That happens to be made by pole 2. And then transfer RNAs are synthesized, of course, by pole 3, and most of them are also made in the nucleolus. So it's a busy, busy place. And in addition, there's a bunch of little pieces of RNA, small nucleolar RNAs, and small nuclear RNAs synthesized here. And we'll meet them in their different roles as we go along. So, how do you make a ribosome? So the big story we're telling now is ribosomal biogenesis. How does a cell make a ribosome? Well, there are genes for ribosomal RNAs. Just like there are genes for proteins, there are genes for microRNAs, there are genes for ribosomal RNAs, there are genes for transfer RNAs. So the ribosomal genes, as I said, you can imagine this DNA loop is actually assembled in the nucleolus. It's a piece of DNA sticking into the nucleolus. And that gene then is transcribed by polymerase 1, and what happens is a large precursor RNA is made. Now you can see different shaded regions, right? So what that means is that some of these regions are going to turn into functional RNA and some are going to be cut out or in processing. And the next step is chemical modification of this RNA. So here you see this pseudo-uridine, that's what this refers to, and 2-O-methylation, the little red dots occur. What's this story about? Well, you know in RNA there's uracil, right, is the base. But in most RNAs, all except messenger RNA, there are pseudo-uridines that are formed. And we think they're probably functioning in providing information for secondary structure. It's not entirely clear. But it's a common feature of ribosomal RNAs and transfer RNAs. And so the way this works is, again, in the nucleolus, there are riboprotein complexes whose job it is to catalyze this formation of pseudouridine. We won't get into the chemistry. Basically, it adds an extra nitrogen onto the pyridine ring is what it does. And that's called pseudouridine, probably involved in structure. And similarly, there's a methyltransferase activity associated with one of these little particles, and it modifies this structure. Now, the next step is the pieces that you don't need, the gray stuff, is cleaved away. Okay? Now, let's not get this confused with splicing that we're going to talk about later. It's a different process. It occurs with different enzymes in a different place. But what happens is there are simply nucleases that come, ribonucleases that cut out the gray parts, and what you have left with are the precursor molecules for the 18S component of the ribosome, the 28S component of the ribosome, and the 5.8S. And all of this stuff then gets complexed with another piece of RNA and proteins to form the final ribosomal particle. Okay? So we're showing a nice little linear RNA molecule, but you understand from the lecture the other day, that ain't the way they look, right? They fold up, they have stem loops, they have secondary, tertiary structure, which is probably what the pseudouridines doing. And eventually they form a globular particle in two different sizes that we're going to see are necessary for translation. Okay, so we've built the pieces of the ribosome, shipped them all out to the ER, waiting on messenger RNA. So where do we get the messenger RNA from? Well, that's a process catalyzed by the RNA polymerase II. Now, if we want to place some emphasis medically, functionally, biochemically, POL2 is arguably probably the most important and interesting polymerase in this whole story. Now, of course, if you work on RNA for a living, what I just said was ridiculous. But I think in terms of medical terms, diseases result mostly from defects in protein function, and this is how you make, this is one of the first steps toward making protein. Okay, so how does all this work? Well, on genomic DNA, POL2 has to know where to start, right? The structure is really important in this whole process. What's the beginning of the coding sequence? What's the end of the coding sequence? What's the beginning of the transcriptional start site? Where's the end of the transcription process? And what's shown here are some DNA sequences that are pretty well defined and pretty famous. And a lot of genes in humans have these features. Okay, so what are they? Well, one is called a TATA box. And the sequence of the TATA box is here, T-A-T-A-T, and TATA box. Okay? That's one that you should remember the name, TATA box. Should at least recognize the sequence. You have to memorize the sequence. But if I give you a story about TATA box sequence or some mutation in the TATA box sequence, this is what I'm talking about. So why do we care about the TATA box sequence? Well, in a lot of genes, not all genes, but very frequently genes that are induced, genes that are expressed under certain conditions, right? You need some inflammatory gene. It gets turned on. It's induced. You need, during development, more myosin to make muscles. That's turned on. As opposed to what are called sometimes housekeeping genes. For example, a lot of the genes of the glycolytic and Krebs cycle, they're needed all the time in every cell except blood, erythrocytes and platelets. And so those genes are generally transcribed at a pretty steady rate in most cells. Many of those, quote, housekeeping genes or maintenance genes often do not have a TATA box. They're called TATA-less genes. So how do we know in those genes where this whole process starts? Well, there's another site here, this GC box, that probably starts the process of transcription. Okay? So let's go back to TATA genes, TATA box genes that are inducible. How does all that work? Well, there's a process, a series of proteins that are going to bind to the TATA box to tell the RNA polymerase machinery where to start. Okay? And we're going to step through this pretty quickly. So here's the genomic DNA. Here's the TATA box. You can't really see it, but it's labeled. And this was that INR site, which is the actual start of the transcription. So the pre-mRNA is going to start here. But the purpose of the TATA box is to get the proteins all lined up in register, to get them into the right place. So there's a TATA box binding protein that binds to the data box. That brings in some of what are called the basal or transcription factors. In this case, transcription factor 2. That means this is a general transcription factor for polymerase 2. You'll see the code TF3. That's a general transcription factor for polymerase 3. Got it? See the code? Anyway, to make a mRNA, some of the general transcription factors then assemble along at the right place. That's the secret, at the right place. That triggers the assembly of the RNA polymerase and a slew of hangers-on, which we don't have to memorize all the names of these things. The purpose of all this is to get the polymerase in the right place to begin the synthesis of the RNA. Now, once this is in the right place, helicase activity begins to open up the DNA. Okay? Other transcription factors will come into play, other more gene-specific transcription factors. A lot of the general transcription factor machinery kind of falls away, and then synthesis will begin. So what are these other transcription factors that I'm alluding to? Well, it depends on the gene. It depends on the cell type. And again, we'll do more of that tomorrow, talk about the specifics. But the principle is, in order for, once the general transcription factors get everything lined up and ready to roll, then there's a question of, well, go or no go. Do we make this gene or not? Okay? If the cell doesn't, quote, need that gene, then typically signaling pathways that control the expression of other transcription factors have either turned them on or turned them off as needed. Okay? If they're present, what happens then is you get this complex machinery formed that then promotes the synthesis of messenger RNA. It essentially induces the RNA polymerase to go forward and to begin reading the code and synthesize the transcript. Does that make sense? Okay. More specifics tomorrow. All right. So, after initiation, then you got to elongate the polymer. And so that's done by, I think, a fairly familiar process here. You got nucleotides coming in, ribonucleotides coming in, sequences being read, and the messenger RNA shown by the little blue thing coming out here is synthesized. Here's the 5' end, and it gets longer and longer and longer. Now, here's an important structure, the C-terminal domain. The reason this structure is important is the polymerase uses this structure to assemble sets of proteins that are really important for further processing of this transcript. Like what? Well, for example, like capping. So there's a set of proteins called capping factors that stick to the 5' end, and they're going to make a chemical cap on the end. And I'll show you details of that in a minute. There are also proteins that ride along that perform splicing. And what that means is removal of introns and then allowing exons to be fused together to get a mature transcript. And then eventually, once the enzyme comes to the end of the sequence that's going to be transcribed, it has to stop, and so there are 3' end processing proteins. And we're going to show that the 3' end gets polyadenylated, binds up with proteins, and gets it prepared to then be shipped out of the nucleus. All right, so how much of that detail do you have to understand? I want you to understand the basics, that during the process of elongation, this C-terminal domain carries along proteins that are necessary for capping, splicing, and then tailing, processing of the tail. So what do I mean by capping? And why does it happen? Here's just a summary of what we just talked about. And this is what's meant chemically by capping. So just to orient you, what you're looking at, this is an RNA strand, and you know that because these are riboses, right? These are, rather than deoxyribose, here's a 2' hydroxyl, so these are riboses. Here's the bases, purine base, pyrimidine base. Now look what happens here at the very 5' end. There's a purine, it's either an A or G, and what these capping factors do as the message is emerging is they catalyze the synthesis of this 7-methylguanine cap, 7-MG. Now, if you look at it, you go, well, wait a minute, it's upside down. And look at this weird bond. There's a 5', 5' set of phosphates here, rather than 5', 3'. Okay? So this enzyme has catalyzed, this actually set of enzymes has catalyzed this funny reaction. And what it does is it changes the structure of the end of this molecule. And in addition, there are methyltransferases that also add some methyl groups, which fundamentally changes the end of this molecule. So why do you want to change the end of this molecule? Why not just leave it be? Well, 2 reasons. One is it protects it from nucleases. Remember, the cell and the environment's chock full of nucleases, endonucleases, exonucleases, ribonucleases, DNases. And their job is to clean up any nucleic acid that's wandering around unchaperoned and off the reservation, so to speak. So to keep this from happening with RNA, the cap is blocked. And so you get longer-lived RNA and you get more protein out of the deal. And in addition, the cap is a structure that's absolutely required for the formation of this sort of blob of proteins, okay, which is necessary for translation. So there has to be a particular set of proteins shown here bound to the cap in order for it to be processed further. Now, what this loop structure and these other proteins are meant to tell you is that the transcript, once it comes out of the polymerase and it's gapped and spliced and polyadenylated, does not sit there as a naked molecule. It is actually rolled up, it's bound with proteins, and it's bound with proteins that protect it from nucleases and also proteins that signal it's time to ship out of the nucleus out into the cytoplasm. So that's what this is all about. All right, so what's the deal with splicing? Bacterial genes don't have introns. We talked yesterday about operons, right? Sets of transcript that are head to tail, head to tail, head to tail, and they make sets of proteins in a coordinated fashion. In contrast, human genes, the transcripts have introns and exons. Okay? Exons having the coding sequences, introns sort of intervening sequences. Remember that? Introns have sequences that do not code for protein. And in order to get a mature transcript to make protein, you got to get rid of the introns, and that's a process called splicing. Now, this is a little complicated, and I want you to understand the concept of splicing and, more importantly, the function of splicing rather than the chemical details. I'm going to have to show you the details for it to make sense and give you a concept. But here's the functional relevance of splicing, or one famous example that a lot of students last year didn't remember and missed on the NBME shelf. So don't forget this, okay? Because it hurt my feelings that they missed that. But anyway, here's the story. Tropomyosins. Tropomyosins are structural genes in proteins. They bind to actin filaments. They're involved in muscle contraction and cell migration. Okay? So here's the deal. There are 4 tropomyosin genes. Right? There are 40 different tropomyosin proteins. So how does that math work? The answer is alternative splicing. In other words, from those 4 genes, you get a whole bunch of different proteins. And the way you do that, the way the cell does that, if it's a striated muscle cell, skeletal or heart muscle cell, it processes the alpha tropomyosin, the pre-mRNA transcript, so that it picks only a subset of all the exons out of the total. And it makes the striated muscle version. If you're a smooth muscle cell, you make the smooth muscle version. If you're a fibroblast, you make the fibroblast version, and so on and so forth. Okay? So how does the splicing occur? The splicing occurs by, it's actually kind of a cool catalytic reaction of the RNA that's aided by proteins that put the RNA in a particular shape. So what you can envision the pre-messenger RNA, which would sort of go along all the way across here, has exon sequences, and they're numbered, 1, 2, 3, 4, 5, how many ever. There's 50-some in the dystrophin gene, for example. And then there's introns, also numbered, 1, 2, 3, 4, 5, 6, 7, 8, etc. And there's a particular sequence that I'm going to show you in a minute that defines the beginning and the end of the intron. And there's characteristically an adenine molecule at what's called a branch point. Okay? Now, this adenine molecule is a busy little character. What happens is that the RNA essentially folds up into loops. The intron's loop, this adenine, it's actually it would be adenosine, attacks this end of the intron, cuts it. That forms this structure called a lariat, right? Lariat. And then this free end attacks the other end of the intron and cuts it out. All right? So you're sitting there thinking, wait a minute. There's lots of A's throughout this sequence. Why doesn't it happen here or here or here or somewhere else? And the answer is, in part, because of specialized proteins. Okay? And the reason the specialized proteins and the positioning of all this is really important has to do with the sort of a biochemical principle, is that the 2' hydroxyl in RNA, most of the 2' hydroxyls in RNA, are highly chemically reactive. Left to their own devices, if you mix them up in dilute solution, they'll go and cut up RNA. Okay? In fact, some types of RNA in cells will actually catalyze the splicing all by itself. No proteins, no nothing, just the RNA. It's catalytic. And the reason is this 2' hydroxyl is highly reactive. And in this particular situation, to form this lariat, what happens is this 2' hydroxyl from this adenosine will then catalyze this reaction, and you'll get a loop formed, okay, with, in this case, you can see a G. Now, the loop doesn't form just anywhere. It forms at this junction. So how do you know where this junction is? Well, there are what are called consensus sequences that have been discovered. Sequences that say this is where the junction is. We don't have to learn you don't have to memorize the details of this, but the junction at this end is typically GU. The junction at this end is AG. That's a very common, and that's where the reaction occurs. But again, how does the cell wind this thing up into this loop-like structure? Because it's a big molecule. It's not going to do that just folding up in solution. And the answer is proteins, and riboproteins. The U family, U1, 2, 3, 4, 5, and 6. So what they do is they assist in the initial folding. That brings the A in close proximity to this junction, right? Then catalyzes the reaction that I just showed you. And some of the U proteins fall away, and some of them come in and then catalyze a further shift that then allows this free end to catalyze this hydrolysis, and boom, the lariat is removed. So what happens to all this stuff? Well, the lariat typically is degraded, but not always, because there are actually some microRNA genes that are coded within introns. I realize that's a complicating factor right now. Most of it's degraded, but some of it is actually processed through microRNAs. And what happens to the message? Well, it gets translated. And we'll show you how that works in a few minutes. Good so far? So again, back to the purpose of this. Why do we care? We care because this splicing mechanism is a way cells can make multiple proteins from the same gene. We also care because there are a number of genetic defects in splicing processes that result in disease. We care because mutations in splice sites can either block normal splicing, which will screw up the protein, or we'll create a splice site, which will screw up the protein, you see, and cause disease. So this is both the source of normal variability in protein production, but it's also the source of pathological changes in proteins in disease. And in this example, a famous example, which I know you're going to remember now, is tropomyosin producing different isoforms. This is the alpha form, tropomyosin gene 1, producing isoforms in different cell types by splicing out different exons and introns. Okay? Is that clear? Does that make sense? Sort of? All right. So capping is going on, splicing is going on, and they're actually sort of simultaneous. Okay? But at the end, termination has to happen to stop the whole process. And there's a particular sequence here, AAU, AAA. This is essentially a stop sequence. It says, okay, transcript is long enough, knock it off. And there are a couple of proteins that, again, that ride along this C-terminal domain of the polymerase, shown here, and they induce the cleavage of the transcript. Okay? So it's cut off, and now what happens to it? Well, now it has to be further processed for shipping. Basically it has to be packaged for shipping out of the nucleus into the rest of the cell. And one of the steps is a process called tailing. And what tailing involves is the catalyzation of poly-A tracks. So, in other words, A sequence. And that requires a poly-A polymerase. And when that happens, that triggers the binding of poly-A binding proteins, whose job it is to protect the structure and allow it to be transported out into the cell. And this is a cartoon version of what that would look like. So here's the transcript out here. Here's the stop sequence. Here's the poly-A tract. And here are the poly-A binding proteins. Interesting thing about the poly-A tail. The length of the poly-A tail has profound influences on how long this transcript is going to live. Okay? Big, long poly-A tail, the transcript has a long lifespan. As it shortens, okay, and after it's been shipped out into the cell and uncoded and translated and so forth, the poly-A tail eventually can be shortened, and then once it's too short, then that triggers the whole transcript for destruction. So the lifespan of the molecule is a function of the poly-A tail. And that's why this is important to happen. The lifespan of the molecule dictates the concentration of the transcript available for translation, which controls how much protein is made in a cell. Okay? Make sense? All right. And that's what I just said. More protein. Now, in terms of the shipping, transport out into the cytoplasm, the poly-A binding protein binds to the poly-A tail. That associates with these capped binding proteins. And again, there's other proteins not shown on here that are coating this molecule, protecting it. And this whole complex is then recognized and transported through channels in the nuclear membrane out into the cell for translation. Okay. Now, you thought we were almost done. We are. But what can happen rarely, and sometimes in a medically important way, is that the RNA transcript can actually be edited, can be further edited. So it's already been synthesized, it's been capped, it's been tailed, it's been spliced. Probably about the same time splicing happens, though. Some transcripts, few, very few, some transcripts can be edited. And what that means is their sequence, their base sequence can be changed. A variety of ways to do this. They're complicated, some are kind of unknown, but there's one that's important medically and that's worth knowing about. And that's editing of the transcript that codes for a protein called ApoB100. So what ApoB100's job is to carry fat molecules around. So when you eat a meal, taking the fat gets digested by lipases in your gut into smaller and smaller pieces. And those lipases then bind and get taken up by the lining of the gut and transported into the blood. They go from the blood, go to the liver, they get processed and transported back out into the blood, out into the tissues, taken up in the tissues, used for energy, stored for energy, and so forth. All right. So you're thinking, all right, blood's aqueous, fats are fat, oil and water don't mix. So how do we do all this? How do we transport this stuff? And the answer is with proteins, lipoproteins. So the lipids bind to proteins at various stages. And you'll learn in some detail later on in cardiovascular how all that works and why that has influence on heart disease. And it's an important story. The story right now, though, is we'll keep it simple, is in the liver, what the liver needs to make is a particular form of this protein called ApoB100. And so the entire transcript gets processed and translated into a protein that has these two domains here, again, involved in lipid transport. And that gets incorporated into a structure called a very large, low-density lipoprotein. Okay? However, in the intestine, right, which is where the fat went in the first place, is a different story. You need a different protein. You need slightly different processes. But the same transcript, the same primary transcript is initially made. That's an important point. The same transcript is made, but then it's enzymatically modified at one nucleotide. One nucleotide. Okay? A C is converted to a U by removing an amino group. And you think, well, wait a minute, we saw this reaction. We did. We saw this reaction in DNA, right? Where you can get hydrolysis of that amine to a U, and that creates problems in DNA. Well, here, this is an enzymatic deamination in RNA, the purpose of which is to create a new protein. Why does it make a new protein? And in this case, you can see the ApoB48. 48 means it's smaller than the 100. Why did that happen? Why do you get the smaller version here than you did there? Anybody know? After the editing, you get a smaller protein. Why do you get a smaller protein? UAA is a stop codon, kids. So instead of an amino acid at this point, right, it makes a stop codon. So you make a stubby protein. And the stubby protein is made in the intestine, not in the liver, and it's incorporated into chylomicrons. Pretty cool, huh? Okay. So like I said, RNA editing is actually quite rare, but there are some famous examples, and this is one potentially shows up on boards because it controls expression of very important proteins in lipid metabolism, which have profound effects on cardiovascular health, blood vessel wall health, atherosclerosis, heart disease, which is the leading killer in the Western world. Okay? So, it's all RNA editing's fault, I guess, in the end. All right, so we have a transcript. Yay! So what do we need to remember about this? You need to know these terms. You need to know the architecture of a transcript. As in test questions, you will see these terms. You have to know what they mean. What does it mean when I say the 5' cap? What does it mean if there's a defect in the genes that code for the enzymes that make the 5' cap, for example? What's the 5' untranslated region? Okay, and the answer is that's the piece before the start codon. Okay? What's the start codon? Well, that's where translation starts. What are exons? Well, they're coding sequences. What are introns? Well, it's a sequence that does not code for proteins that is removed during splicing. Okay, there's a stop codon, we just met, and a 3' untranslated region that has a poly-A tail. Why is this structure important? Well, we don't have time to go into the details, but there are There is sequence information in the 3' untranslated region and the length of the poly A tail that have dramatic effects on the lifespan of the transcript. Therefore, can have dramatic effects on the amount of protein and the time that a protein is made. So why would the timing of a protein be important? So think about kind of two things. Think about development. Okay? If you have a cell, fairly rapidly growing little bundle of cells, and some of them have to migrate off and become part of the brain. So they have to turn on the right set of adhesion molecules, motor proteins, actin cytoskeleton, matrix proteins, so that they can go wander away. Once they get to where they're going, they've got to stop. So typically what happens is those proteins are stopped being expressed, and a different set of proteins turns on. Well, how does that magic happen? Well, partly by events in the 3' UTR, partly by transcriptional control, partly by translational control. All right? How about inflammation? You get person gets infected, you get swelling, you get puffiness on your skin, it hurts, it's painful, and eventually it resolves. Well, the immune system senses that, activates cells, migratory cells that again migrate in toward the infection, but they have to stop at a certain point. They have to then stay there. They have to then sort of kill whatever's causing the problem, and they have to clean it up. And all of this takes orchestration of the right sets of proteins at the right time. And in the case of inflammation, this whole 3' UTR story is huge. That's a major way that cytokines and chemokines and pro-inflammatory proteins are turned on quickly for whatever time is needed and then turned off. Okay? So to understand that and the medical importance of that, it's worthwhile knowing that a little something about this architecture. Okay, we're almost done for this first part. Let me finish up with pole 3, and this will be quick. So all we're going to say about it is that it transcribes the transfer RNAs. More detail about that next hour, and some small nuclear RNAs. Now, we met the small nuclear RNAs. They're some of the characters that catalyze, that become part of the ribo nucleoproteins that catalyze pseudouridine and 2-omethylation. Okay? And again, they live in and around the nucleolus. Some of the tRNA synthesis actually occurs out in the nucleoplasm as well, but catalyzed by pole 3. So what did we learn? We learned all about pole 1s, 2, and 3. We learned a little bit about ribosomal RNA, ribosome biogenesis. We learned a lot about mRNA and messenger RNA transcription, processing, capping, tailing, splicing, editing, and then coding with proteins for shipping out into the cytoplasm. And then once we get out in the cytoplasm, what's going to happen? Translation. All right? So that's what we'll talk about next time. Let's take about 10 minutes, and we'll come back and get started on the next round. Okay. How are we doing? Questions? Is that clear? Don't get too bogged down on like the nitty-gritty details of the names of all those little proteins. Big picture. Know the names of the processes. Be able to recognize a story if it's about capping, tailing, or splicing. Okay? All right? Okay. Take a break.